and the director of TGen North, which is the infectious disease arm of the nonprofit Translational Genomics Research Institute. He oversees a number of research groups that are working on the development of analytical tools to detect and characterize infectious diseases such as tuberculosis, valley fever, MRSA, and others. His team at TGen also provide genomic services to CDC and to our local health departments. He has published 115 scientific papers and chapters on epidemiology, disease ecology, genetics, and microbiology. He also has 24 patented inventions, including a valley fever molecular assay that just recently received FDA clearance. How long did that take? He received his master's in microbiology from Colorado State University and his PhD in biology at Northern Arizona University. Previously, he was a biologist for CDC in Fort Collins. He was the Arizona Department of Health Services chief for the Office of Bioterrorism, and he also served as Arizona State epidemiologist. He has worked in federal, state, and local government and has started for-profit and nonprofit businesses. Dave sits on a number of local and national boards as well. Recently, he led an initiative that ultimately established Flagstaff, Arizona as America's first STEM city. So, please help me extend a warm welcome to my friend and my ex-colleague, Dave Engelthaler.
I think I need to turn this one off so that there's not feedback. Is that correct? All right, can you guys hear me? If it's too loud, let me know, because I tend to talk really loud, so I don't usually have a microphone. Thank you, Roxanne. Roxanne and I have known each other for a very long time, and she's always one of my favorite guests to come down and visit. She's always trying to set up some type of connection or entertainment, and is always inviting me, so that's how I come down to Phoenix. But again, I'm Dave Engelthaler. I'm up in Flagstaff. I'm part of TGen, the Translational Genomics Research Institute. Has anybody not heard of TGen before? Okay, good. Well, that's not good. TGen's been around for 17 years now, I think, in the Valley. So the Translational Genomics Research Institute came out of the Human Genome Project of the 1990s. You remember there was this race to sequence the human genome. And in the end, it took about $3 billion in about 11 years. But the human genome got sequenced. We were able to spell out every single letter in the human DNA. But the person who was running the intramural program at National Institutes of Health, Dr. Jeffrey Trent, decided we can't just be sequencing. We need to do something with this information. We know a lot more now about genes associated with cancer and cardiovascular disease, that we need to translate these findings into new diagnostics and new drugs, new technologies for more accurate and better patient care. So he came out to Arizona to found TGen as a nonprofit that's focused on translational research, translate these great outcomes or these great findings. So at TGen, we do a lot of basic science, basic discovery, but we're always, eye on the prize is always making an impact on primarily on human health. Really, how do we best use technology, next generation technology and science to better the lives, primarily of patients. And so the focus was a lot on chronic disease, cancer, cardiovascular, autism, Alzheimer's, anything that people think of as chronic or complex diseases that have some type of genetic association. Our group in Flagstaff was founded just over 12 years ago. Last month, we just celebrated our 12-year anniversary of opening our doors. We're infectious diseases, so we still use we have that same mission. We use that great technology, but it's really on infectious diseases. My background, as Roxanne mentioned, and as our connection before, is in epidemiology and public health. And with infectious diseases, That's really important because it's about populations. It's about how people interact and spread diseases, how diseases and pathogens move around in the natural environment. So we can't just be thinking about the patient, we think about populations as well. So we have a really strong public health focus, and we do a whole number of things that I would love to talk to you guys about, and at some point maybe in the future can talk about some of the other really cool and exciting research that we do. I also talk about zombie apocalypses, which is just a way of thinking about infectious disease epidemiology from a more fun perspective. Today I want to talk about some of the research that we've been doing associated with some fungal diseases, some really important fungal diseases, but how we can now really tie next generation science to an understanding, a historical understanding of the movements of people, of a geologic historical understanding, the movements of animals, to anything from animals back to the continents. And so I kind of put together a little bit of talk. It's going to be a little bit of free form, but it's in this Prezi format. This type of format, I'll try not to go too fast because people can get seasick a little bit. I'll try to keep it entertaining as much as possible, but I am going to talk science. I apologize. I'm going to get a little geeky, and I'll try not to get too far in the weeds on some of these things. But just let me know, throw things at me, whatever. I'll move it along if I got stuck somewhere in the weeds. Sorry, Roxanne. What time do I have till? I can go 5 minutes or 5 hours. So sorry, guys. Okay. Okay. Great. Yeah. Somewhere in there. All right. So I won't do a full hour, but I certainly can. So we'll start off with this map. I love maps. I really love this map. This is a mural at the National Geographic. I actually had to pay for the rights to use this map, but now I can use it however I want. So I use it in multiple discussions, and I've used it also in publications too. It's a 1922 sailing, our map that shows some old sailing routes, and gives kind of a cool feel for how people really think. We're starting to think about the world. There's both hemispheres, but today we're just going to stay in the Western Hemisphere. And you guys probably saw the title of my talk, Pathogen, Paleo-epidemiology from Pangea to Present. So I apologize for the alliteration and all the syllables in that, which is a little silly. But it's really, paleo-epidemiology. Epidemiology is the study of diseases, the things that cause diseases, how diseases move around. And paleo, thinking about really into the geologic past. And then Pangea to present, that's pretty obvious. The other part to this is, I say, a tale of Cocci and crypto. So and if you guys can't see some of these things, I will certainly read and try to describe what's up here. But there's more seats up here, you guys, if you want to come up. I don't bite, although it might be a splash zone. I get a little excited. So the two fungi, Cocci and crypto. So Cocci is short for Coccidioides. That's the valley fever fungus. Hopefully everybody's heard of valley fever. If you're in the valley, you should know about valley fever. Cryptococcus is another fungus that's primarily found in tropical parts of the world, and it's mostly in the environment. People get sick from it, and there's a lot of things we can learn about studying the genomes, the DNA of it now, that kind of unlocks some of the secrets of the past for these organisms. So this is where we're going to start, and these, as I mentioned, this is primarily a discussion in the Western Hemisphere. Coccidioides, the valley fever fungus, is only in the Western Hemisphere. Crypto is found in multiple parts around the world. But let's start in a part of the world that we're all familiar with, and I call this the land of Cocci. So the desert Southwest, this is where valley fever is really important. It is mostly important in Arizona and California. Arizona, Maricopa County, more cases here than anywhere else in the world. So this is the hot spot for Cocci. Although it might not be the birthplace of Cocci, I'll show you that. And so if you guys don't know, Cocci, and I'll describe the fungus a little bit more, but this is the natural environment for Cocci. This is where it lives and thrives. In a number of the deserts of the Southwest and other parts of the Western Hemisphere, but the Upper Sonoran Desert seems to be the hotbed for Cocci, and it's because of the organisms it infects, which is primarily rodents. and in a very diverse ecosystem like Upper Sonoran Desert. Humans also live in the Upper Sonoran Desert, and so there are multiple ways that we come in contact with the environment around us. And Cocci, being an organism that's in the dust or in the dirt, can get up into dust and we can get exposed to it. But it's not only found in the desert southwest. It is in desert areas, but Cocci is actually found in multiple parts of the Western Hemisphere. So in some of the hotter, drier places in South America, some places in Brazil, Paraguay, Argentina, even some places in Venezuela and Central America where you have really hot, dry soils. So that's important for us to know, but for me it's really interesting, like how on earth did it get down there, or where did it come from? I have colleagues down in South America. They were adamant that Cocci came from South America and it was shipped up to North America. They really wanted that point of pride, but I've actually had to kind of prove to them they were wrong. That's not exactly what happened, and we'll discuss that in a minute. But Brazil is not really thought of as Cocci, the land of Cocci. Brazil is really the land of what I call the land of crypto. So Cryptococcus actually loves the Brazilian basin, or the Amazon basin in Brazil, but it's found in many tropical and subtropical regions. The fungus itself is one of those fungi that survives on decaying plant matter. So you find it in decaying trees or dead parts of trees in the soils where trees have, and other plant material is starting to degrade. That's where crypto really survives best. And we also know that humans come in contact with this environment as well and can radically change the environment, but also come in contact with the organisms within the environment. And like Cocci, crypto is not just only found down in Brazil. It can be found in other parts of the world, and strangely showed up in the Pacific Northwest. So this tropical pathogen shows up Vancouver Island, other parts of British Columbia, Oregon, and Washington. And so for me, again, I want to know why and how that happened. And was it just recent? The first cases were actually found in 1999, so seemingly pretty recent movement up there. And I think it's really important for us to understand how these organisms move around. And like I said, through the power of genomics, we can start to understand these, not just the organisms and the diseases they cause now, but what happened before and how did they get to where they are. So let's go back to Cocci. I'm going to go back and forth a little bit between these two fungi just to kind of move the stories along. But just to talk a little bit more about the fungus itself, and I know you guys can't see this, so I'll just describe some of this. So this is the life cycle of the Coccidioides fungus. It really has two separate life cycles. It's called a dimorphic fungus. It actually has two different morphologies. In the parasite, it's more like, in the parasite phase, when it gets into a mammal, it's more like a yeast. You get these spores into your lungs, the lungs set up shop, and they just essentially start to replicate in the lungs. And then you could develop a lot of little endospores. They rupture, they can go to other parts of the lung, and just keep that cycle going. But if you're a mammal that's susceptible, you likely get Cocci and die. If you're a rodent, you die in the soil, and now you've got a body that will decay in the soil, and then the fungus can get out of the body and actually set up shop. It's a real hotbed of a fungus right there where the animal died, either on the dirt or down in a burrow. And then it sets up its saprobic life cycle, where it really actually survives on nutrients from the soil and becomes a mold. Essentially, it'll get the hyphae like a mold, a bread mold, and then those hyphae can have individual little spores that, if the soil gets disturbed, they break off, they float around in the air, you breathe it in, and you start the other cycle. So that's kind of the basic background on Cocci. And if anybody has questions along the way, please feel free to ask. Oh, I'm getting a little washout here. If you can't see, that's North and South America here. The colors represent where Cocci's been found, and this is a tree. I'm going to show you some genetic trees and some genomic trees along the way. I'll try to describe them, but without getting too much detail. This is a colleague of mine now, when he did some work at Berkeley about 15, 16 years ago, really established that there's two major groups. So you can see that the tree is showing two major groups, which we now are two different species of Cocci. He called them the California group and the non-California group. So you can tell he's at Berkeley. But really, the vast majority of cocci is in this non-California group. That's what's in Arizona. Is that a hand up in the back? No, it's not. So this is the Coccidioides, which causes the actual disease in mammals is called Coccidioidomycosis, which is why I don't say that very often. It's just got a couple more syllables. But no, this is a fungal disease and is not associated at all with cattle disease. So anyway, so these two different groups, which are now two different species. The other thing that he found out is that there's some what we call phylogeography, meaning that depending on where you're at in the world, you have your own population. So you can see this population structure that's really kind of established by different regions, which makes sense. That's the way most organisms are if they're not interbreeding. So we don't see, it looks like distinct populations are showing up in South America versus Mexico versus Arizona and California. But this is earlier DNA fingerprinting and really needed to get into genomics. I'm going to be really sad if we can't see these trees. I wonder, you do have the lights off. Are these? Oh. Yeah, it's washing out. I mean, it looks, you guys want to come up? It looks really good on my screen up here. No, that's all right. Hopefully I don't have too many of these. What you would see if you could see my screen, we really still see those two major groups. But now I'm showing the two different species. So that one group up on top there, that's Coccidioides immitis. That's what was called the California group. And this group here on the bottom is Posidacei, Coccidioides posidacei, which is pretty much everything else. And so these are our two species that we can now see really by looking at the whole genome. The other thing I want to point out, and this is pretty interesting, you can't see these really long branch lengths on this tree that separate these two species. That means this is incredibly old. The branch length is really essentially a measurement of how many mutations occur between the different groups. And that length of this branch length is estimated somewhere between 1 million and 5 million years for the most recent common ancestor to both of these, which is somewhere out here. We don't know what that ancestor is. There's lots of missing links in fungi, but it's a really old fungus, and actually fungi are really old anyways. So that's a really robust separation. If we start to look at these populations within each of the species compared to where they show up on the map, that starts to allow us to understand, okay, what's different about these populations and how diverse are they, and give us some understanding of how old they are. So the green here there's lots of tree branches in here that you can't see. That's all the southern Arizona, northern Sonora group of Posidacei. The blue is all the Texas, Mexico, South America, and it's a lot less diverse. These are much more closely related to each other, even though they cover this very large geographic expanse. That orange or peach is the Phoenix area, very little diversity within that group. And then there's the yellow-orange above that. That's the population we find in Guatemala, and almost no diversity within that group. And then the red is the California group, really only found Central Valley, Southern California, Baja, and then one population up in Washington. These will all be important here in a minute for this story. One of the other things that we can do with phylogeography, we do a lot of our phylogenetics, which is that studying of the populations, is trying to understand how old they are. We can use advanced Bayesian statistics to really get a pretty accurate, as accurate as we can get, molecular clock to understand how far back then these branches are really telling us. Put this onto a clock and tell us how old these populations are, which ones are older, which ones diverged out of other ones. And so what we see is those groups I was just talking about. The Arizona clade, of which the Phoenix clade is just a subset. All these other ones are found down in the Tucson or southern Arizona region. I call it the Tucson clade. But this is the oldest, seems to be the oldest population, whereas you have the Phoenix clade, which is a very specific group, then Texas, Mexico, South America, then in Guatemala. We're going to put these back on a map here. I promise you, there's continents there. You can't see them. You guys can probably see this timeline. If you can't see the numbers in the way back, this is present This is 200,000 years before present, 400, 600, 800,000 years. Those would be important benchmarks. So the Sonoran population, or the Arizona Sonoran population, we think is about, specifically about 750 to almost 800,000 years old. That's the oldest population. The Phoenix population, or the Central Arizona population, is actually about 100,000 years newer. So the original population that we can identify is really southern Arizona. The Central Arizona comes out a little bit later. The Texas-Mexico the Texas-Mexico group really showed up probably just before the Phoenix group. So there's some movement a little bit to the southeast from southern Arizona. And then you get the South American group. That's coming out about another 100,000 years after that, or 150,000 years after that. So there's some delay in the movement of it down to other parts of the world. The Guatemala population is much newer, probably about 175,000 years ago. And so then what I want to know is, okay, if we can understand, we can time when these things move, is how did they move? Why did they move? And so by knowing this knowledge now, we can do a couple of things. One, these are distinct populations. So they're not continually coming out of there, which means it's not likely something like wind blowing it down. Because if it was, it would be continually blowing it down. We'd never see the difference between these populations. They're very distinct. So we know that's not likely the case. We also know it wasn't humans moving it down. Humans move a lot of organisms around the world. But this is, at the earliest 150,000 back to almost 800,000 years ago. First humans on the Western Hemisphere about 15,000 years ago at the earliest, at least from what we understand. So it's not human movement. So the next logical thing to think about is animal movement. This is a disease of mammals more than it's not a disease of the soil or plants. It is a disease of mammals. And so likely has something to do with animal movement. And so we can think about timing, and you guys can't see, but it says GABI1 or GABI3 and GABI4. So GABI stands for the Great American Biotic Interchange. So if you guys don't know what that is, it was actually a really critical thing that happened for the fauna of Western Hemisphere that actually had to occur at a specific time. So if we think about the connection between the two continents, we go down into the Panamanian Isthmus, that actually did not occur, that land bridge did not occur until after the continents shifted enough. There was some subduction, some push-up of land masses, and it wasn't until about between 3 million and 6 million years ago that these two continents actually started to have a land bridge. And this land bridge that we have today, and this is, you can see Panama here, actually really didn't occur until about 1 million years ago. After that, the land bridge is formed, and then you get enough of an ecosystem going that actually supports then the movement of the animals between here. So going back 3 million years ago, there were some animals that moved down, and it wasn't until this land bridge formed and you had this really good ecosystem that you had mass movements of animals occurring between the two continents. So you had this Great American Biotic Interchange, and we had major fauna moving both directions, right? So it wasn't until this happened that we could get opossums and armadillos and porcupines up into North America. But more importantly for Cocci, it wasn't until this bridge that happened that wolves didn't move down into South America, weren't able to move down to South America. Other mammals that really could carry large Cocci loads. Cats move down, but cats don't typically get Cocci very often. It's pretty rare. But one of the big ones that gets Cocci is camels. So we had massive camel populations. We had mammoth populations. We had our own lions, our own zebras. All these fauna died amazingly about 8 to 10,000 years ago. Once humans started to populate the area, we wiped out all these tremendous megafauna. But the camels that moved down 1 million years ago became the alpacas and the llamas of today in South America, and they're highly susceptible to valley fever. That could be a really good target animal that brought valley fever down into South America, as well as wolves and others. Okay, so we're going to jump back over to crypto now. And we'll come back to Cocci, I'm sure. But I want to talk about crypto as an organism and then how it's how it moved around the planet as well. So Cryptococcus is a fungus also of the environment, but really seems to be found, as I mentioned, more in tree holes and in decaying plant matter. There is one species of crypto that actually is spread by birds, and typically pigeons, and it's moved all over the world because pigeons have moved all over the world. But primarily, we'll focus on this Cryptococcus gaddii, which is really a tropical, subtropical fungus. And not to focus, I don't really focus on the human disease here, but you can get those yeast cells that are in the soil up in the air, breathe those in, cause a pretty massive infection in the lung, and then some of these species will actually escape the lung and cause a mostly encephalitis, meningitis and encephalitis, so a neurologic disease. And this is actually one of the most important pathogens for HIV-infected individuals in Africa. They first die of TB or they die of Cryptococcus. Those are the two big things that HIV-infected individuals in Africa die of. Here's kind of a boring tree, but it's an important one. This is done by another colleague of mine in Australia, but again, really older DNA fingerprinting technologies and some older molecular clocks have shown that the major species in Cryptococcus diverged millions and millions of years ago. So the original estimate was about 49 million years ago from the major branch. That's actually now that we have better genomics and better tools, been increased to about 100 million years ago that these major groups separated. So this is a very old organism. And then within these groups, so the Neoformans group up there, that's the one that's spread by pigeons. The Gaddii group is the one that we're going to focus on. There's actually individual species within these groups. We thought this was a species. These actual groups are individual. These represent millions of years ago, so even these separated out probably tens of millions of years ago. And so, which is interesting for our story. We're going to focus in on this species primarily because that one seems to have moved out of the tropics and has gotten into a couple other areas like the Pacific Northwest. Now we're able to replicate this with the genomics. Again, you can't see the trees very well, but these branch lengths, again, kind of point out how old these organisms are. And again, tens of millions of years within some of these groups. And some of these groups actually are fairly new. We would normally, medicine treats them all the same. But in reality, there's a lot of things going on here that we didn't understand until we could actually start to tease it apart with genomics. And the age of it is one of those things. I don't even know what my next slide is, so it's a surprise to me too. Oh, okay, great. Pangea, of course. That's even in my title, and I forgot. So one of the thinkings about how organisms can move around, the animals can move around the environment, wind, humans, but also the possibility of plate tectonics. And so a couple of great researchers, including Arturo Coccidioides, who's the lead author on this, from Harvard, put out a paper suggesting that it was the actual breakup of Pangea that may have been related to the speciation event that caused the two big Cryptococcus gaddii, Cryptococcus neoformans, that separation, which I think makes a lot of sense. I was a little disappointed when I saw this come out because just a couple years previous to this, for some of my dissertation work, I was thinking about the same exact thing, but I was thinking about those species that are in the Cryptococcus gaddii group with those obnoxious names, VG1, VG2, VG3. And looking at the world as to where those show up, at least the names are up there, you can't see the continents, but the different colors represents those different species. And I was looking at where they are endemic, or where they seem to have been up for a very long time, and I always thought it was interesting that VG2 primarily in South America, the Brazil area, but also up in Senegal in Africa, the only place in Africa where it exists. Or the VG4 species is almost completely in South Africa, but also up in India, the only place in Asia, in the Asia-Europe landmass where it exists. Because if you go back to Pangea and you look at where those are, Senegal was what was connected to Brazil. And India was connected to South Africa. India completely away from Eurasia, right? When the Pangea broke up, India hightailed it around and met up, crashed into Asia, formed the Himalayas and Mount Everest and all that. But before that happened, it was connected in this area. So I thought that was interesting. I actually had, ended up publishing on that too, that idea and thinking about it. But the more I think about it is, that happened 100 million years ago at best, that final breakup of Pangea. These populations are only tens of millions of years old. So probably something else is what actually caused those separations in the locations. And one of the things I'm thinking about is the movements of ocean currents around the world, which is possible. What's interesting is that Cryptococcus as a fungus actually can survive in salt water. We don't know how important salt water is or the ocean is to its life cycle at this point, but it's been a number of studies to show that it can survive. There is movement of water between those areas that we're talking about, in between South America and Africa and Africa and up to Asia, but it's going in the wrong direction for where I would have thought that would have happened. Now, of course, the water currents have shifted over the years, and some things have caused that to happen that we might get to. It might not be the best way for this to have happened, because the other thing is too, if it was the water currents that had moved it from South America magically from here all the way up to Senegal, it would have likely shown up in other places as well, and it's not. So it's probably not that happening, or that's not the main driver of those kind of speciation events. But there's one other thing that I think is really important. Probably the most important thing maybe that ever happened to our planet was the KT event. The meteor, or asteroid, I just don't know what to call it, that caused the Chicxulub crater that essentially wiped out the dinosaurs. It didn't just wipe out dinosaurs, it wiped out most life on the planet. Over 70% of species disappeared after this event on the planet, of animals and plants. This event not only changed the course of history for the planet and caused then volcanic eruptions in other parts of the world that allowed for all this death to occur. These animals die. What happens when the animals die? Was that? Yeah, fungus. Fungus takes over. Now, fungus can just have a heyday. This is the age of dinosaurs turned into the age of fungi. And so much ability for diversification and movement and growth that I think that probably at that time period is when we actually had effect on a whole bunch of different species of fungi that we have today. It also caused so much material to get into the air, blacked out the sun for at least 6 months, but 6 months to a year, that essentially ended up killing off most plant life. And so even with all of that decay too, so there's just more and more opportunities for the fungi to take over. And that's in the tens of millions of years ago. So I think that was a really important driver. Of course, Arturo Coccidioides, that same guy who beat me to the punch on the plate tectonics, also published on the dinosaur theory as well. But I think it's a really good theory. I think it's really important. So thinking about crypto, we also know that it's more recently moved around the globe. And those are things that maybe we can pinpoint a little bit better. So as I mentioned, up in the Pacific Northwest, there was a couple of cases seen in 1999, the early 2000s, and it was about 2006, then there really seemed to be an outbreak occurring. In this VG2 group of Cryptococcus that I was talking about, there's 3 different subgroups in there, what we call A, B, and C, really clever nomenclature, of these groups, or of the species showed up seemingly out of nowhere and then started causing disease. We jumped in with some genomics and again developed these family trees for those groups, and we were able to see there's actually was some diversity. Remember, these branch lengths mean mutations between individuals. If it just happened, they would all look the same. There'd be no evolution having occurred, but there has been some evolution. It's little. Those branch lengths are not very long. Those are at most tens of SNPs, tens of mutations, versus those other branch lengths we were looking at were with hundreds of thousands of mutations between individuals. So they're still really closely related, but we're able to, in order to understand then, what does that mean in context of the whole world, I worked with collaborators around the world. We gathered every single isolate of this VG2 species that we could find and sequenced them all up. And then developed this type of a tree. Just helps us understand relatedness and who comes from what. Where those different strains came from, all roads led to Brazil, essentially. There was a Brazilian isolate on every single lineage that we could find or closely connected to it. So we most likely think that this essentially came out of Brazil, this VG2, at some point. Wherever it is in the world now, it started in Brazil. And so that was important for us thinking about these three lineages up in the Pacific Northwest. Published on that too. The other thing we could do is do this advanced statistical analysis to see really how old that is. So one of those groups, what we call the VG2A, we did this beast Bayesian analysis, and this is really computationally intensive. This is like developing 4 billion of these individual trees and finding where coalescence actually occurs. When did they first really have a most recent common ancestor? And it looks for this group, it's about 88 years old. So it's not yesterday, but it's not that old. So that's something we can get our hands on and try to figure out why did it show up there 88 years ago. And then we looked at all three groups. And so, yeah, so about 90 years and about 70 years for the other two groups. So pretty close to each other. And these tools can be off a little bit. The fungi don't follow the rules. They do have all sorts of strange reproduction. I mean, you think humans are weird, man. Fungi are really weird. I mean, where it's all, they can have sex with different, quote unquote, a different type of sex. It's a different type of terminology. They can have sex with themselves. They can have sex with multiple at the same time. Maybe it is like humans, but that means that you get a whole lot of strange diversity can occur. So things can happen where the molecular clocks get off a little bit, but they're pretty closely connected to about 70 to 90 years ago. So that's, we published on that too. So then that allows us to start thinking, okay, what happened? Well, how did it get up to the Pacific Northwest 70 to 90 years ago? Well, of course, I'm looking at my sailing map here, and that's how I think, well, humans definitely had something to do with this. It's not animals moving up. It's not some kind of just natural movement up, because we would see it elsewhere along the way. We did not find it elsewhere between Brazil and the Pacific Northwest. It got straight from there. And 90 years ago, we were not flying much in the way of, I doubt there was a single plane flight from Brazil to Vancouver 90 years ago, but there was some shipping. In fact, more and more shipping started to occur, but before 90, 100 years ago, the only way to really ship is to take one of these routes, and you can actually follow it on this map. There it is, all the way up to the Pacific Northwest. But that rarely happened. There was not a whole lot of shipping occurring. It was really treacherous coming down around the bottom of the continent, so there wasn't much going to that side. It was mostly shipping to the eastern coast of North America. But that all changed. We look at Panama and Central America. Remember this from Cocci, and that land bridge needed to form for Cocci to get down to South America, but it took Teddy Roosevelt and near slave labor for about 10 years to carve this big old trench through Panama, which then allowed for ships to really move from the east coast of South America to the west coast of North America much more easily. And shipping actually really started to occur in mass between the continents that way. And in fact, if that's the case, if we can think about shipping, we can start to think about where maybe it really originated out of Brazil. And going back to this tree, we look at one of those subgroups that actually showed up in the Pacific Northwest, and this particular strain is actually from Brazil. All these other ones are pretty much have a tie to the Pacific Northwest. This one was a patient in Recife, Brazil. Recife is a major port city of Brazil, and in fact, they did not start international shipping until after the Panama Canal was opened up, and then they started shipping actually directly to California and up into British Columbia as well. Or they started at least movement of materials and started doing people shipping, kind of tourism, but being able to go visit other parts of the world. Actually, as I mentioned, I like this map. This was a paper I published this map in to kind of describe then what we thought of as the Teddy Roosevelt effect. That's why we're thinking 88 years ago at most. It couldn't have been too much further because there was no way for material to get, or for the fungus to get up into the Pacific Northwest. And it took that event for that to happen. But there's still got to figure out what is going on, what was moved up there, how did it get up there, why are we just seeing cases, in the last decade or so? Well, I actually did go look at shipping manifests from Recife and from other port cities up to Vancouver from the 1920s and the 1930s, and I couldn't find a ton of material, but I did find that they did ship things like rubber. There was textiles and some other things going from Brazil up there, but nothing that says we're pulling something out of the environment and taking it up there, not like dead trees or lumber, because Pacific Northwest, they got all the lumber that they want up there, so they're not getting stuff out of Brazil. Just more like finished products and other materials. So what I wanted to do was actually get a better understanding of what may have happened up here. So what this map is looking, this is Vancouver Island, and this is the rest of mainland British Columbia. And all the red dots represent human cases of this cryptococcus that occurred over the last 20 years. The blue dots indicate animal cases, or veterinary cases. And a couple of these dark blue, you can see those are porpoises. There's actually infections in sea animals, which is also really interesting to me. This fungus is, people were trying to figure out where there's rain and then drainage back out into the ocean that may have been infecting, as porpoises, dolphins, a couple whales, sea lions, otters. So mammals, sea mammals are really susceptible to this for some reason. But the other thing is these are all along the coastal forest. You can't really see there's this pink area. That represents coastal forest versus really the thick mixed conifer further inland. So most of it's along these coastal forests, which to me I'm thinking this is just mostly everything close to the water. So my feeling is that what got shipped was water from these port cities. If you have contaminated water in one port, they pick it up as ballast in boats. This happened a lot with early sailing boats. Now there's a lot more rules and regulations about this. But you pick up water to help with your balance depending on the load that you're shipping. And then when you get to the next port city and you offload your goods, you offload your ballast as well. And so a lot of microorganisms can be passed along with this. Well, Cryptococcus, interestingly enough, as I mentioned, can survive in salt water. This is the fungus. It has a capsule that it creates. And what's just been understood is that it can actually control the size of its capsule, and that actually affects its buoyancy. Of course, this is another paper by Arturo Coccidioides, who I keep following around the globe. I think he thinks I'm some kind of fanboy, which I actually am. I think he does amazing stuff. But he's not only smarter than me, he's way cooler than me, because when he put this paper out, he drew this picture and tweeted it to let everybody know his paper came out. And I would never think to do that. But I think the ballast water is actually a pretty interesting idea. And that way, certainly sea mammals can get infected, and it gets along the coastlines. We got to think about why is it up into the forest then, and why is it causing human cases and animal cases? Well, you remember this tree, and this is just representative of a number of trees that I've looked at and trying to understand the population up there. This is about 100 years ago, right? So 90 to 100 years ago when we think the most recent common ancestors. But there's this other node, this other, what we would call a vertex of one of the four vertices that show up in this tree, is occurring about 50 years ago, 50 to 60 years ago. And we don't exactly know why. It just seemed to be an anomaly within the tree, but other trees have shown. What I think is that there's two diversification events. There's this original one where the population showed up, and then something else caused then the population to go wild and get a lot of diversity. And so then, if that's the case, so you can see I'm starting to do some hand waving. This is what we do in science sometimes. So, just because you try to pull a story together, then we got to go disprove the story. And if you can't, then you've added more evidence to your overall story. So about 50 to 55 years ago, what would have happened to have that come up out of the water then, if it was there maybe for a few decades, surviving and thriving in the water in the nearby coastal systems? Well, to my next part of the story then is we got to go to Alaska, because almost exactly, 55 years ago from last month, the Great Alaskan Earthquake occurred. And in 1964, this was the largest earthquake ever recorded in the Northern Hemisphere. 9.2 on the Richter scale. Nothing else was seen in the Northern Hemisphere. There's one that was slightly larger down in Chile. But in the Northern Hemisphere, the largest one, at least that we've ever recorded. And so that, I mean, which caused just massive devastation, coastal cities. This is downtown Anchorage, so you could just see that this kind of devastation could really affect the environment. And so one of the things that an earthquake does, especially along the coast, is to cause tsunamis, what they called tidal waves back then. And the tsunami effect was felt as far away as Hawaii and registered even down into Oceania as to how massive the effect was. But you could see up there by Alaska, that red area, that's where the biggest effect was. It was so massive that Seward, Alaska, a small coastal town, multiple accounts of a 320-foot wave. I mean, I can't even imagine that coming in. I mean, that's like a bad Hollywood movie, right? That happened up there. But it also happened along the coastline, not that big. But you get that much water movement, you're actually going to push water up into the tree line, which is what I absolutely think happened in the Vancouver area, Victoria Island, that you would have actually had, if you had living organism in the water, that gets all pushed up in the tree, and most of that's going to die. But something like cryptococcosis actually survives and thrives in a forest. So quite a bit of hand waving, I know, but it's for me it's an interesting theory, or I shouldn't say theory, hypothesis of how then an organism could end up in a different part of the world causing a disease where it's never existed before, and following this type of a pattern geographically. So I actually, we're planning some studies to go in and do some sampling and see if we can show this. One of the other things that gives me some evidence is there's this Port Abierto here in Vancouver Island. There was some cases up there, both animal and human, and people thought, well, that's just the progress of the infection over here. But this channel got incredibly devastated by that tsunami, so much so that town actually almost got completely wiped out. 30-foot waves came up all the way at that channel. You can imagine the pressure forcing all that water flow and wiped out most of the towns, or most of the town. And there's been some dolphin cases over here as well. So I think that the organism actually probably got pushed up that way somehow. And I think the tidal wave, the 64 tidal wave it's my favorite explanation anyways. Okay, so before I wrap up, I want to go back to Cocci just for a minute, because we didn't even talk about the California species very much, the C. immitis, or the Coccidioides immitis. And we're also up in the Pacific Northwest. Strangely enough, Cocci has found a home there. Now this is actually southeast Washington. This is what they call the Tri-Cities area. That's Kennewick. Kennewick is famous for, anybody know Kennewick? Kennewick Man, yeah, very good. So the oldest, most complete fossil of human in North America was actually found there in Kennewick. But also in Kennewick is a dry, deserty area of Washington, and there's a ATV park. Somebody had fallen, got a leg wound. They ended up getting Coccidioides in that wound. And so it made sense that it must have been something in the soil. Epidemiologists go on ground, they dig up the soil, they actually can get Coccidioides out of that. We did all the genomic testing and showed that these were identical. And so this tree here is the California group, the Coccidioides immitis. What's circled up there is the Washington group. So there's been multiple cases now found and multiple soil samples that are positive. We sequence all of that. They're almost the same. There are small branches in there to say that this thing's not brand new. We actually estimate somewhere between 600 and 6,000 years ago. That's a pretty large range, but it wasn't new. And for me, I thought that was really interesting. 6,000 years ago, Kennewick Man was 8,000 years ago. I think it's quite possible that you had some California populations or people that got exposed in California go back up and settle and maybe died with valley fever and set up a new population up there. Total hand waving because I don't have any proof around that. But we did do the genomics and show that this is a very distinct cluster out of the California group, this immitis group. But thinking about the California group, That's that group up there at the top on this slide that you can't see, so we'll go back to this. But, and here's the map you guys can't see either, but this, we talked about the Postadaceae, the California group is up there in the purple, and let's put that on the time scale. So this C. immitis, Coccidioides immitis, the most recent common ancestor we can find for all the California group is about 375,000 years ago. That is much newer than what all these other populations are occurring. And which is okay, it probably wasn't part of that Gaby event because it's moving this direction. It's possible that the movement of animals between continents was somehow involved, but that's not really what's probably driving what was happening up there. And then the next population is the Southern California, Baja, Mexico, about 200,000 years ago, so newer population than what was found in Central Valley of California. And then this very, you can't see that color, but the Washington, much newer, like I said, about 600 to 6,000 years ago. So what happened then that may have been driving the population diversification of this California group? Well, about 700,000, little over 700,000 years ago, was the draining of the Central California Basin. That's where most of this cocci is found in California, is in the Central Basin. And why that's important is, I mean, we'll go in our way back machine, back to the Miocene. 15 million years ago, there was no Central Basin. That was the coast of California. This is before the uplift of the San Francisco Ridge, which occurred in the Pleistocene about 3 million years ago. We could actually start to see really the formation of what is now is the West Coast. But it formed this very large saltwater sea in California where we would normally, that's San Joaquin Valley, the rest of the Central Basin. And it wasn't until about 1 million years ago that actually started to drain out. The salt water started to drain out. You have primarily, you have a solid land bridge there now, so there's not any ocean water coming in. But the Central Basin was still not, it was still underwater, but it was all under fresh water. That's because the Sierra Nevadas were still in the Ice Age. Sierra Nevadas, the glaciers would melt, fill that up with fresh water, and then that would drain over thousands of years, and then you'd have continual climatic shifts of which the building up and shrinking of the glaciers and this melt. So then you had all of that occurring, and that final drainage didn't occur till less than 700,000 years ago to get what we have now, which is that Central Basin, one of the most fertile growing basins or growing areas in the world because of the geological formation. But it also now is this great fertile growth for Coccidioides. In fact, the valley fever was named after San Joaquin Valley. That's where the first cases were identified. It's not the Valley of the Sun, where most cases occur. It was actually there. And so, there's a few other things. The only difference that we can ever find between the California group and everything else is that the California group, immitis, is halophilic. So it can actually survive better in saltier environments, which makes sense. That soil out there is much saltier than the soil in the Sonora and in the Chihuahuan deserts. And so this is most likely, we understand where it is. This is most likely the timing and the why of it occurring in California. Okay, that's almost as much hand waving as I could do about this, but I could do a little bit more. It's to think about, okay, so we think we've learned a lot about where these organisms came from, but let us think about where they may be going. We know that humans are going to continue to move these and other organisms around the globe. This is such an important part of public health right now, is understanding the global setting to understand what we're going to have in our own backyard here. And we see it all the time. Influenza moves every year and moves pretty quickly around the globe. And some of these new emerging diseases pop out of nowhere. We're always worried about Ebola when we should be worried about getting our kids vaccinated for measles. And sorry, I don't know if I can't imagine there's too much pro-measles in the group. I'm kind of anti-measles. But we also got to think about what's also changing on our planet. So climate's changing, animals move around, but the other thing that's changing is that these continents have not stopped. They're still moving. Continents move at about how fast your fingernail grows every year. So annually, you can imagine how fast your fingernails will grow. That's how fast the continents move. And so there's been some thinking about what's going to happen 50 million years into the future. The thought is Africa and Europe are going to smash together. There's going to be a brand new mountain range that's rising up there. But that'll be one large land mass based off of the movement of the tectonic plates now and the modeling. We don't know for sure, but then 150 million years from then, Australia and Antarctica become a single landmass, and the two sides of the globe actually get closer to each other, at least continent-wise. And then 250 million years ago, we're back to Pangea. This is actually being called Pangea 2, which is not very novel. You can come up with something better than that. But the thought that you're going to have nearly one large giant landmass, which means that animals will be able to move and carry their pathogens very easily. I don't know that humans are going to be around here. Hopefully we'll be someplace else, but 250 million years from now on planet Earth seems a little crazy. But we can start to visualize what that means, and I won't go too far into that, but certainly continual movement of animals and plants and their pathogens. But that's not where, I don't think it stops there. As I mentioned, humans might not be there, but humans are already in space, right? So International Space Station, we've had people living up there for a couple decades, not all in a row, but some people do live up there long periods of time. We have people continually going up there, and what do? People were covered in microbes, and so we bring our microbes into space. And so there's been a lot of studies looking at all the microbes on the International Space Station, everything that we've brought up there, and how they're evolving. And some of them evolve differently. Some of them become a little bit more resistant to radiation because the radiation is a lot higher, and so they can survive in a high radiation environment. Microgravity is affecting evolution and a few other things. They're developing some new functions. And so I got to throw this up there because one of these guys, Venkat, who's one of the leads at the Jet Propulsion Laboratory, funded by NASA out of Caltech, and Marcus Calcum is a colleague of mine as well too. We just wrapped up a study where we just handed off to JPL some analysis of material both from the space station as well as material that's being prepped to send into space as part of a larger project. We're totally excited about this. Don't know what it's going to mean for human health, but what we're trying to do is not totally mess up the space environment. So JPL is designing all the instruments that are being sent on the Europa Clipper. The Europa Clipper is actually going to send a probe to Europa, one of the big moons of Jupiter, and we know it's covered in an icy crust. This probe is actually supposedly to break through the icy crust to get down what's below, which we don't truly understand. It might just be a big methane ocean. But what we don't want to do is carry our own pathogens in there. We're going to probably contaminate, but really what we're going to do is confuse. And we don't want to be finding DNA pathogens or DNA organisms on other planets until we fully understand those planets or moons, just other bodies. So there's some cool far out stuff that we get to work on with the technology, but it won't end there. We will take our pathogens and our microbes with us, and so I think the last thing that we can think about is actually where else fungi and these other pathogens will emerge. It will be on the moon and it will be on Mars. This is no longer pipe dream. It looks like images from some sci-fi books from a couple decades ago. These are actually images I just downloaded from the SpaceX website. This is they're planning within 10 years to have some type of bases set up on the moon, as well as having at least one human visit to Mars. But it's not just SpaceX; it's also Blue Origin, Jeff Bezos' company, and Virgin Galactic, and others. And this is definitely happening. It's going to be, and it'll be happening in most of our lifetimes. Probably within the next 10 years, we're going to see some of this, and we will, no doubt, we have to bring our microbes with us because we can't survive without our microbes. So what's going to happen? I don't know. I don't know that Cocci is going to show up, although Mars looks like a nice Cocci environment landscape. It's probably a little cold for Cocci, but certainly within our living quarters and other areas, we've already seen it on the space station. We'll see it on the moon and Mars too. And that is as much hand waving as I could do. So be happy to take any questions.
Now you see why I stay in touch with him.
Any questions? Yeah.
Yeah, I think I heard that there was like some fungus growing on like Curiosity on Mars already.
So we don't have, obviously don't have any samples to do the testing. And I actually have not seen the details. I've heard that as well too. And I don't think Curiosity, the probe that, or the rover instrument, whatever, I know that JPL is sending, the next one, is actually going to have DNA sequencers on board for that exact purpose, to truly understand what might be. Otherwise, we can only pick up certain signals. I think that the current one has a mass spec or something like a mass spec that helps understand the different chemicals, and the thought is that there may be, but I don't know for sure.
I saw just the other day that I believe it's in Colorado. They uncovered a fossil bed from the day, the KT boundary.
Yeah.
I assume you're going to study beneath that and above it to check on the fungi?
Yeah, so I think that was I think it's Wyoming, and it's where they had found this. And maybe they've seen some in Colorado too, but they're definitely, but certainly these layers, these KT layers, have been found all over the world. What was recently found is, I think it's Hell's Creek in Wyoming, was actually the remnants of the tidal wave that occurred from the Chicxulub crater. That far inland, which is really remarkable. They're finding fish and other sea life that would have occurred down in the Baja or the Gulf of Mexico, actually up into the Hell's Creek area. So that's, to me, that's fascinating. But you're right, it's looking at these cores that go through these KT boundaries in a number of areas, and doing some ancient analysis. So DNA does not survive very well in the environment by itself, and we're not at a point where we can pick up some molecules, but not enough to do sequencing in some of these really old things. So it's mostly looking at fossils, and fungi are soft body, don't really leave much in the way of fossils, but there are some clues, some pieces of evidence, amber and whatnot, where actually they do pull fungi out. And yes, we will be cloning that and growing that, there's no doubt. When George Church back east is still planning to grow a mastodon, so it's not just Michael Crichton's imagination. We'll be able to study it that way by getting the DNA and growing those organisms.
I wanted to ask, when you said that about people and animals moving the disease, do you think that it ever is able to pass directly from animal to animal or person to person, or is it the decaying bodies and it starts over again as spores?
Yeah, so some pathogens definitely, we know, will spread from human to human. And there are some fungi that do, but the vast majority of fungi don't. They typically, or the vast majority of environmental fungi don't. They need to have, the growth phase that's appropriate to cause infection. So we don't have movement of Coccidioides between animals. It's likely, or it's possible when we have transplants, and we have seen that with transplants. We've done some investigations because transplant doctors, is there a transplant doctor in the house? These guys are crazy. We know patients are dying of certain infectious diseases. They will still take organs out of that patient and put it into the new one. They know they're putting in the pathogen, but they say, we'll just give the new patient, the recipient, antivirals, antifungals, antibacterials, and then hopefully they'll survive, because it's more important that they get a new heart than it is for them to have a clean heart. I get it, but we actually do spread organisms that way. But naturally, no, we don't see it with Coccidioides.
Question that might take us off topic, and if it does, then
What was our topic?
I know. The newer fungus that they're finding around the world that people are dying of, and it seems to be a different mutation in each location.
Yeah.
Anything on that?
Yeah, so Candida auris. Candida is a type of fungus that it's really a pathogen that can be spread between humans. This Candida auris, we didn't even know about this thing before like 7 or 8 years ago. And then all of a sudden seems to be showing up primarily in healthcare centers, so it's a hospital-acquired infection. in multiple parts of the world. And you're absolutely right. We've done some of the sequencing. We've worked with CDC on this to try to understand these populations, but it's still an anomaly because it's like these things just popped up, and they're really new in these areas because there's not a whole lot of mutations within each one, but they're very distinct from each other. So it's not easily spreading from one place to another, but they're just popping up in the healthcare system. And there's theories out there as to why that may have happened. It may have occurred in some product developments that different hospital centers have used, and then so at some point there was this early spread, and then it's allowed to diversify, and now we're starting to see those patients showing up, and that's why they all look distinct in these different areas. But we don't know. It's fascinating, highly drug resistant, very difficult to treat. So yeah, it is the fungal superbug of our time.
So recently my dog started limping, and I took her to the vet, and he said, oh, we're going to do some blood. He did some x-rays, and he said, we're going to do blood tests. And I said, what are you testing for? And he said, valley fever. Now, she was not having any lung problems, just this limp, and it came back positive. She's on antifungals. And I want to know, how does it settle into a leg rather than into lungs?
Yeah. So the vast majority of the way people get infected is the same way dogs get infected, is that they breathe it in, and then once you get this infection in the lungs, it can actually essentially escape out of the lungs and become multisystemic. It can go to different parts of the body. It can go to the bone, it can go to the blood, it can go to different organs, it can go to the brain. And in humans, 50% of the time it gets out of the lungs, it goes to the brain. So it's very serious, that disseminated, what they call disseminated cocci. And sometimes it just disseminates to one place, and it'll set up shop in a joint or in a bone. And that sounds like what may be happening in your dog, although it may be in other parts of the dog too. It's not easy to find everywhere there's a disseminated infection like that. So yeah, in dogs, they are probably more susceptible than humans. It's estimated that 1 out of 3 naive dogs will get infected within the first, I can't remember if it's within the first, I think it might be within the first 3 years. Yeah, get infected. But 33% of all dogs that are new to the environment, whether they're puppies or they've come from other parts, will develop some type of a cocci infection within 3 to 6 years. I can't remember. But they're very susceptible, and unfortunately, there's not great tests for dogs, and there's not great treatments either. So, good. Yeah, good. Yeah. So the fungal drugs that we have do not kill the fungus. They just keep it in check, prevent it from reproducing. But the fungus is still alive, so they rely on the immune system then to get rid of the fungus. So if it's a strong enough immune system, then the drugs will work pretty well. The problem is that in humans, we don't give the drugs to people unless they have a serious disease, and they typically don't have a serious disease unless they have a weak immune system, whether that's from immune-suppressed drugs, infections, old age, diseases, like diabetes or something else that lowers the immune system. So it's not a good system. We need better drugs. We've been doing some drug development work in that area as well too, because of that particular problem. We need something that actually kills the fungus, because people have to be on drugs for the rest of their lives, and people and dogs. Dogs are people. Any other questions?
This is quite simplistic, and I'm embarrassed to ask it. What is a fungus a mold a fungus, or are they two distinct entities?
Yeah, so the mold is essentially a growth phase of a type of fungus. So typically fungi are microscopic fungi are thought of in molds or in yeasts. And so the yeast will develop as individual cells, and the molds typically have like the hyphae, like the bread mold kind of a thing. And so it depends on the type of fungi. Fungi are so incredibly diverse if you think of a tree of fungi, you could think of one branch and one sub-branch off of that, and that would be all all vertebrates in the world, not just mammals. And then you have the rest of this diversity within fungi. So the fungi are incredibly diverse, and they have lots of different lifestyles. And so some of them actually have both lifestyles together as a mold and a yeast. Some, mushrooms are fungi. That's a completely different life state or style as well. So to answer your question, yeah, a mold is a fungus is a mold.
I wanted to ask another question. I work in the medical field, and we had a continuing education about cocci, and we were told that Filipinos especially are inclined to have cocci.
Mm-hmm.
And in general, we find at the hospital a lot of times Pacific Islanders, African Americans, more so than Northern Europeans. And I wondered, did you ever do anything related to the Native Americans who would most likely be living amongst this for the longest? How did they experience cocci?
Yeah, so that's a great point, which we don't fully understand, but Filipinos, by far, the largest, and then African Americans and some other populations of color, typically not have higher rates of getting cocci, but higher rates of getting disseminated. So the much worse form, and it is a problem within Filipinos that's not necessarily shared with all Asian populations, which is interesting. And so there's a number of genetic studies actually that are occurring right now to understand it's typically some mutations that occur in some populations within specific immune cells that probably is allowing for this dissemination to occur more frequently. Native American populations, interesting, I've definitely had that same thought. We don't see higher rates there. And that actually makes sense on an evolutionary timeline that maybe if the original populations came over, may have been more susceptible or whatnot, but there's probably been enough interaction that you would see some positive selection for things that would prevent dissemination and death. The time scale is still very, very short in the overall time scale of cocci, right? So the last 15,000 years versus the last million years. So cocci probably hasn't evolved a ton to counteract anything that may be selected for in these populations. And I don't think cocci cares at all about humans. I think humans are just an accident, right? But humans themselves may have some adapted features. We've certainly seen that with a lot of other pathogens around the world.
Okay, and I think this will be our last question.
Okay, then I'll shut up.
Last but not least, is penicillin a fungus?
So Penicillium is a fungus, and Penicillium is a type of fungus that we've used to develop penicillin, which is one of the compounds that's made by that fungus. Penicillium also makes other compounds that have been used for other drugs, and a lot of fungi, because they survive and have to be in this war all the time, they have come up with this diversity of compounds to survive different environments, to compete their competitors, to fight off other types of infections. That is one of the richest sources for new drugs and new drug targets. Everything from statins to new antibiotics. So we have many, many lines of antibiotics that have come out of fungi. Penicillin is the original great one.
Well, thank you.
Thank you, guys.
Thank you so much, David. We have a tradition of mugging our speakers, and so if you come back often enough, you'll have a whole set of them.
Oh, awesome.
you.
Thank you. Appreciate it, Roxanne.
That was great.
Oh.